All right, my name is Sabine Lindler, and I'm going to tell you about my Pinhead internship at the Maine Medical Research Institute in the Epigenetics and Obesity Laboratory. So I applied because I'm very interested in nutrition. I read the book Eat to Live by Dr. Joel Fuhrman, and ever since then I really changed my lifestyle and I was really passionate about nutrition. So I wanted an internship related to that. And also, I wanted to challenge myself and do science in the real world instead of just getting the watered-down kid version. And so Pinhead placed me in Scarborough, Maine, at the Maine Medical Center Research Institute, and they put me in the Epigenetics and Obesity Lab, so the obesity tied into my interest in nutrition, and I'll explain the experiment in just a minute. My mentor was Dr. Robert Koza and Mark Kozak on the Pinhead staff. His father, the TSCC staff, his father was Dr. Koza's right-hand man for years, so he used that connection to hook me up, which was great. And I also worked with Rhea, who was Dr. Koza's wife, and she also worked in the lab. So the purpose of this experiment was to observe the epigenetic mechanisms of obesity in experimental mice fed a high-fat diet. And so you might be wondering what epigenetics is, and before my internship, I was wondering what epigenetics was too. So basically, epigenetics means that two organisms can have the exact same sequence of DNA and can live in the exact same environments, but there can still be variation because their genes are being expressed differently. So you can think of it as being two identical pianos with the exact same keyboard, but you can play two very different songs. And because the keys are being played differently, just as the genes are being expressed differently. So epigenetics accounts for the variation in fat mass of these mice, even though they're all genetically identical and they're all fed the same food. So a lot of the experiment involved measuring gene expression, and we used RNA. We found out how much RNA was in the tissues of these mice to determine how the genes are being expressed, because DNA by itself doesn't really do anything, but once it's converted into RNA, it can be used to make a protein, and the protein is what carries out the function of that gene, causing it to be expressed. So if you have more RNA, you have more expression. And so the first thing we did was isolated the RNA. So what we had to do was take a little chunk of tissue, mix it with some reagents, and then we would homogenize it. So basically, it would mash it up and suspend it in solution. And this is a video of the homogenizer machine. And so you'd have the test tube with the tissue in it, and then it would mash it up. And then after that, we would filter out all the other components of the tissue, so we just had the RNA. Then we prepared to put it into a machine called the QRT-PCR, which stands for Quantitative Reverse Transcriptase Chain Reaction. And so we had to prepare the RNA to put on a plate, and the plate looks like the picture in the top right-hand corner. So we used a machine called a nanodrop, which is pictured in the top two left-hand corners, and that uses light waves to read the concentration of an RNA sample, and you only need a tiny drop of sample, so that was really cool. And then once you know the concentration, you can do some simple calculations to dilute all of the samples down to the same concentration. Then you use Excel to make a map like the one pictured below so that you know which sample is in which well on that plate so that you don't get things mixed up. And then you use a pipette. This is a video of Dr. Koza using a pipette to put the samples onto the RNA plate. And there's a lot at stake here because it's very easy to mess up. You have to be very focused because you could, if you forgot to change the pipette tip before loading the sample, you could cross-contaminate and ruin the entire experiment. So you have to be very focused, and it's kind of scary. And then after that, you put the plate in the qRT-PCR, and that causes the RNA to replicate so that you can then find out how much you have. And here's a video of me explaining how we do that.
It's a qubit.
It's telling how much RNA is in here. So it's saying how many nanograms of RNA is in here per microliter. And in this little tube, there's about 200 microliters. So I open this up and I put this in here and then shut it. Is that right?
Right.
Read next sample.
Yep.
And I hit read next sample. And then you can see there that it's 164 nanograms per microliter. So then I just...
Or per mil.
Or per milliliter. Sorry. And then just take it out. So then with that data, we can know how much RNA of a certain gene was in the tissue samples from the mice. And then based on how much RNA we have, we know how much gene expression we have. So then Dr. Koza taught me how to make figures representing the gene expression of different genes that he's associated with fat mass in mice. So I actually made those figures, and it was really cool because until we did that experiment, no one knew that that was true. So I actually helped figure out a new scientific discovery, which is really cool to be a part of, because in school, they just tell you what's already true. But in a Pinhead internship, you can make new discoveries that can contribute to the world, which is really awesome. So now that I've talked about life in the lab, I'll talk about life outside of the lab. So my host family was the Birchills, and they had an 11-year-old daughter and 2 dogs. And it was really nice of them because they let me use their car, and they borrowed a car from a family friend, so I didn't have to bike 6 miles each way to my internship through traffic. So that was so nice of them. I had really great living conditions. And I hooked up with the Scarborough Girls cross-country team. So I had some running buddies, and I made some friends while I was there. My host family had some friends who had a boat, and so I got to jump into the freezing ocean off of it, and that was fun. And then my host family also took me to the beach to see touristy stuff like lighthouses and all that. But ultimately, this Pinhead experience was so rewarding because I got to be a part of new discoveries, and that's what really sets a Pinhead internship apart from just any other opportunity in science for kids, because with the internship, this is the real deal. Like, this is the PhD doing his experiment, and you can't mess it up. It's not like the watered-down version for children. Like, it's a big deal, and so it's really rewarding to know that you did that and were a part of it. So I really enjoyed it. So I'd like to say thank you to Pinhead for this amazing opportunity. I'm so grateful for setting me up with this. Thanks.